Hi students, and welcome to Year 11 Biology and Module 2, Organisation of Living Things. This is video number 21, and we're going to just review what we already know about gas exchange in plants. Specifically, we're going to investigate the exchange of gases between the internal and external environment of plants. Specifically, to help us do this, we're going to try and review what we know or what we've already looked at previously around gas exchange. We want to identify the two important processes. So firstly, respiration. And we know that relatively, when we compare these two processes, the rate of respiration in plants is much lower than the rate of photosynthesis. So the other important process that involves gases is photosynthesis, and the rate in plants is very high. The key gases that we want to think about when we're reviewing gas exchange are water. Water can and is certainly a liquid moving through many parts of plants, but it's also in gaseous form, moving particularly in the leaves. Oxygen, which is also an important gas. It's required for respiration, but it is a product of photosynthesis. And the reverse of that applies to carbon dioxide. Carbon dioxide is a very important raw material for photosynthesis, but it is a product of respiration. The exchange between the external and internal environments are facilitated by, in the leaves, stomata, that is individual stoma, individual pores that are surrounded by two guard cells, and the way that the guard cells behave in relation to how much water they have regulates the amount of exchange that can occur between the internal and external environments of the leaf. In the roots and the stems, we find similar structures to stomata, but these are lenticels, and they're again another type of pore that allows exchange between the internal and external environment of the plant. So we know that one of the important things is that we have lots of air spaces. Air spaces are pathways for gases, and the gases are going to move both into the plant from the external environment, so from the atmosphere, which is the external environment, and they're going to exchange with the internal environment, which is what's happening inside the plant. So we classically find this leaf structure with lots and lots of spongy mesophyll that's allowing a lot of passageway of gases to move between the cells, but also the more columnar and structured palisade mesophylls that are closer to the upper epidermis, and still allowing a little bit of diffusion of gases from areas of high concentration to low concentration. The loose packing of the parenchyma cells provides this interconnecting network of air spaces, and the gases are going to diffuse through the air several thousand times faster than they do through water. And this is why there's a much more efficient exchange through these gas spaces within the leaves than we do find, for example, when they are dissolving in water and being transported through the plant that way. Oxygen and carbon dioxide can reach the network of intercellular air spaces, and then they diffuse rapidly through them. Remember, the important thing about diffusion is that it's moving from an area of high concentration to low concentration. We do look too at some of the factors that affect the rate of gas movement or even the rate of water loss. So water is actually going to come the other way. This is H2O coming the other way. That's going to be affected by how quickly you can build a concentration equilibrium. If you can't, if, for example, it's a hot windy day, if it's a day where there is low humidity, for example, you're going to continue to find more water is going to be released through those stoma from the internal environment of the plant to the external environment. And oxygen and carbon dioxide are also going to pass through the cell wall and the plasma membrane of the cell by diffusion. And specifically, when we're looking at gas exchange, this is the actual way that gases move from these air spaces into the cell or from the cell back out into the air spaces. So we do have to allow for this exchange of gases from some part of the cell or from the continuity that it has with the external environment into and out of individual cells. But that means one of the key structures that's associated with gas exchange are the stomata. And this, as we've talked about previously, involves a very delicate balance between gas exchange and water loss. Because it's important particularly to get carbon dioxide into the plant, but at the same time that the stomata are open, we also have water coming out of the plant, then we have to consider the balance between each of those. If there's sufficient water, the rate of water loss might not be significant. If water is limited, then that may be a critical step in terms of keeping the plant alive. As a general rule, the stomata usually open when the light strikes the leaves in the morning, and then they close during the night. So as the light disappears at the end of the day, then we are going to see the stomata closing. You get a little bit of an idea about those timings from the table that I've got here, which shows some of the differences in osmotic pressure, which has been taken from the BioLibre text, which I've referenced in other videos. And these values you can very clearly see. The highest pressure is around 11 a.m., middle of the day, lots of sunlight. The low pressures are midnight, so when it's darkest, that's when the osmotic pressure is low. And low pressures, because of the way that the guard cells are structured, low pressure flaccid cells are going to collapse in on the stoma and close that pore. It's the nature of the structure of guard cells with radially thickened walls around the stoma that means that when the turgor pressure increases, so as water moves into the cells, they swell, but they don't just swell evenly. And that's the important thing about them. They look more like kidney shapes. So the swelling is actually going to drive them apart from one another, and it's going to open the pore in the middle, and that's going to allow, or at least it's going to allow, an increase in the rate of gas exchange. So when we're thinking about some questions that are associated with this particular area, we want to be able to draw a diagram of a root hair cell which shows the movement of material across its surface, so you can get an idea of that high surface area to volume and some of the reasons why for the exchange of materials, and also some of the changes which might take place in guard cells over a 24-hour period, and perhaps think about how that might have an influence on things like the rate of photosynthesis and also the rate of water loss through the plant. Thanks for watching.